Hello, everyone. Today, we're diving deep into one of the most powerful techniques in molecular biology, real-time polymerase chain reaction, or qPCR. Whether you're a student, researcher, or lab professional, this comprehensive guide will help you understand how qPCR works, its applications, advantages, and limitations. So grab your lab coat and let's get started. Let's begin with a simple question. What is real-time PCR? At its core, qPCR is a laboratory technique used to amplify and quantify DNA or RNA. But what does that mean exactly? Well, let's break it down. PCR stands for polymerase chain reaction, which is a method for copying DNA. It's like making multiple copies of a document, but instead of documents, we're copying DNA. And when we talk about real time, we mean that this copying process is monitored as it happens, allowing us to quantify the amount of DNA at each step. This differs from traditional or conventional PCR, which detects the presence or absence of DNA at the end of the reaction, a method known as endpoint detection. Now, why would we want to do this? Why not just use regular PCR? That's an excellent question. We'll address the advantages shortly, but for now, let's focus on the basics of how qPCR works. The PCR cycle consists of three main steps: denaturation, annealing, and extension. During denaturation, the double-stranded DNA template is heated to around 95 degrees Celsius, which separates the strands. Next comes annealing, where the temperature is lowered to allow small, single-stranded DNA molecules called primers to bind to their complementary sequences on the template. The final step is extension. Here, an enzyme called DNA polymerase starts synthesizing new DNA strands by adding nucleotides that are complementary to the template. The amazing thing is that these steps can be repeated over and over again, doubling the amount of DNA in each cycle. But here's where qPCR shines. In contrast to conventional PCR, qPCR incorporates fluorescent molecules into the reaction mixture. These molecules emit light when excited, allowing us to monitor the progress of the reaction in real time. Now, there are two primary ways to incorporate these fluorescent molecules. First, we have a dye called SYBR green. This dye binds to any double-stranded DNA, meaning it's not specific to our target sequence. As the PCR reaction proceeds and new DNA strands are synthesized, the fluorescence signal increases. The second approach uses sequence-specific fluorescent probes called TACMAN probes. These probes have a fluorophore and a quencher molecule. When the probe binds to its complementary sequence, the fluorophore and quencher are close together, resulting in a decrease in fluorescence. However, when the probe is cleaved during extension, the fluorophore and quencher separate, leading to a rise in fluorescence. Both methods enable us to track the amplification of our target sequence, but using different principles. Now let's discuss how we actually quantify the results from a qPCR experiment. To understand this, we need to introduce a key term: the threshold cycle, or CT value. The CT value represents the number of PCR cycles required for the fluorescence signal to cross a predefined threshold. In simpler terms, it's like asking, how many cycles does it take for our signal to become detectable above background noise? The lower the CT value, the more starting material we had in our sample. For example, if one sample has a CT value of 20 and another has a CT value of 30, it means that the first sample had 1,000 times more starting material than the second. To determine the actual quantity of our target DNA or RNA, we can create something called a standard curve. This curve is generated by running reactions with known amounts of our target sequence. By comparing the CT values of our unknown samples to this standard curve, we can back-calculate the amount of target present. Now that we know how qPCR works and how to quantify our results, let's explore some of the key applications. One major application is in gene expression studies, particularly with RT-qPCR. Remember that qPCR amplifies DNA. So if we start with RNA, we need to convert it to DNA first, using an enzyme called reverse transcriptase. This process is known as reverse transcription. The resulting DNA is called complementary DNA, or cDNA. After this initial step, we can proceed with qPCR to amplify and quantify the cDNA. By comparing the expression levels of a target gene in different samples, researchers can gain insights into various biological processes, such as development, disease, and the response to environmental stimuli. Another important application is in pathogen detection. qPCR is a valuable tool for quantifying viruses and bacteria. For instance, in the case of COVID-19, qPCR has been widely used to detect and quantify the SARS-CoV-2 virus. The ability to precisely measure viral load is crucial for diagnosis, monitoring disease progression, and evaluating treatment effectiveness. Let's move on to another significant application: genetic testing. qPCR can be used to detect single nucleotide polymorphisms, or SNPs, mutations, and copy number variations. SNPs are single base pair changes in the DNA sequence, while mutations can involve insertions, deletions, or substitutions of larger DNA segments. Copy number variations refer to differences in the number of copies of a particular DNA segment among individuals. These variations can have important implications for disease susceptibility, drug response, and other traits. In addition to these applications, qPCR has found uses in various other fields. In food and agriculture, for example, qPCR is employed to identify contaminants or genetically modified ingredients. In environmental science, it's used to detect and quantify microorganisms in soil and water samples. The versatility and sensitivity of qPCR make it a powerful tool across diverse scientific disciplines. Now let's discuss some of the advantages of qPCR over conventional PCR. First, as we've mentioned, qPCR offers precise quantification capabilities. With conventional PCR, we can only determine the presence or absence of a target sequence. Second, qPCR is much faster than conventional PCR. It typically takes a few minutes to complete a qPCR run, compared to several hours required for conventional PCR followed by gel electrophoresis. Additionally, qPCR is a high-throughput technique, commonly performed in 96 or 384 well plates, allowing for the analysis of numerous samples simultaneously. This throughput makes it suitable for large-scale studies and high-demand applications. Despite these advantages, qPCR does have some limitations. One major drawback is the cost. qPCR requires specialized equipment and reagents, which can be expensive. Another limitation is the complexity of the technique. Careful probe design and the inclusion of appropriate controls are crucial for accurate results. Additionally, false positives can occur, especially when using SYBR green as the fluorescent dye. It tends to bind to non-specific products, which can elevate the fluorescent signal. To mitigate these challenges, it's essential to use validated assays and implement proper experimental design and data analysis strategies. As technology advances, exciting developments continue to push the boundaries of qPCR. One notable innovation is digital PCR, dPCR. Instead of continuously monitoring fluorescence, dPCR partitions the reaction into thousands of tiny droplets. By analyzing the distribution of positive and negative droplets, dPCR provides absolute quantification without the need for a standard curve. This increased precision is particularly useful in fields like oncology, where accurate measurement of rare mutations is critical. Moreover, the emergence of portable qPCR devices holds promise for point-of-care diagnostics. Imagine diagnosing malaria with a smartphone-sized qPCR device. It would be a game-changer for outbreak tracking and resource-limited settings. In conclusion, qPCR, or real-time PCR, is a revolutionary technique that has transformed molecular biology. Its ability to amplify and quantify DNA or RNA in real time makes it an indispensable tool for research, medicine, and industry. From gene expression studies to pathogen detection, genetic testing, and beyond, qPCR continues to drive groundbreaking discoveries and advancements. As technology evolves, we can expect even more innovative applications and improvements to this powerful technique. Thank you for joining me on this journey through the world of qPCR. If you found this video helpful, don't forget to subscribe for more science updates. Until next time, keep exploring the fascinating world of molecular biology and the incredible tools that help us understand life at the genetic level. Thank you.